Hello everyone and welcome to today's webinar. On behalf of Protein Simple and our webinar organizer, Cambridge Health Tech Institute, I would like to welcome you to our discussion today regarding development and optimization of capillary electrophoresis methods for quality control of biologics. My name is Xin Jian. I'm the product manager of ice and Maurice systems at Protein Simple, and I will be moderating today's event. Our first speaker, Dr. Chen Liu, received her PhD in physical chemistry from Brandeis University, where she studied ALS disease through protein characterization using mass spectrometry, and developed novel techniques such as HDX monitored by ultra-high resolution mass spectrometry. In 2016, Dr. Liu joined STC Biologics, which provides integrated therapeutic product development. She is now leading the Analytical Development and Protein Characterization Group at STC Biologics. Our second speaker, Krista Edwards, is currently an assistant scientist at Celldex Therapeutics in Fall River, Massachusetts, in the Analytical Development Department. She graduated from the Catholic University of America in Washington, D.C. with a bachelor's degree in biology, where she developed an interest for lab research while working in a cancer research lab during her senior year. And now I'd like to welcome our first speaker, Dr. Chen Liu.
Thank you very much for the introduction, Xin. It's my pleasure to share my Maurice experience today. The first part of the talk is about IC-IEF method transfer, and the second part is about CESDS method validation. STC Biologics is a boutique biotechnology CMC development service organization in Massachusetts, with core competencies in integrating biology with process, analytics, formulation development, and regulatory sciences. STC provides GMP release and stability testing support, which will soon to be complemented by GMP manufacturing, targeted to be commissioned in Q1 2019. Both studies to be shared today were sponsored by our clients and performed on Maurice system. As we know, a single Maurice system can support both IC-IEF and CESDS detection schemes. At STC, we have taken advantage of both. I'm going to first talk about PI variation observed during IC-IEF method transfer of one IgG1 molecule and the following method development. In the second half, I will share the story of CESDS method validation for another IgG molecule. Let's first start with the IC-IEF method transfer. The IC-IEF assay was initially developed for the IgG1 molecule on an IS-280 or IS-3 system. 6 charged isoforms were resolved, including 2 acidic species and 3 basic species, and the pI of the main isoform fell in the range of 8.2 to 8.7. The method was then transferred to a contract lab using a Maurice system. Similar charge profile was observed, however with improved resolution. One additional acidic species was resolved and showed up at the shoulder next to the main isoform. Besides, a significant shift in the pI of main isoform was observed. Its pI was 7.32, which was about 1 pH unit less basic. At that time, STC was brought in as the third testing lab and precipitated in an investigation. The investigation was carried out in an instant, efficient, and organized manner. Rather than being comprehensive, the high-risk and easily accessible aspects were first examined, which are the materials and methods. The material tested at both labs was supplied by the same manufacturer. It was confirmed that there was no unexpected change in primary sequence. The material was stored frozen. Besides, if the protein degrades, it most likely will result in profile change, not only PI shift. We then examined the method. The transfer method was outlined in the bottom left. After continuous communication, it was found that the formulate composition, protein concentration, and focusing time were off. Out of those three parameters, the formulate composition probably contributes most to the PI shift. The reason is, in ICIEF, the reported PI was determined by interpolation from the PI markers, assuming linearity. However, the non-linearity of pH gradient in ICIEF has been observed and reported. The top left graph shows that the grade pH linearity of broad-range formulate was negatively impacted by buffer excipients. Here is PBS. The top right graph shows when mixing broad-range and narrow-range formulates, the linearity of pH gradient was also negatively affected and can be further impacted by addition of PBS. In summary, the reported PI can be affected by many factors, including temperature, additives, usage of different PI marker, and fundamentally, the linearity of pH gradient. Due to the identification of possible root cause to the PI shift and the observation of improved resolution, a DOE study was designed to reconstruct the PI shift and improve assay performance. The DOE study showed that by changing the formulate composition, mixing up to 50% narrow-range formulate, the reported PI for the main species changed from 8.26 to 7.42, which is almost a 1 pH unit difference. It proved that the change in formulate composition can greatly affect the reported PI. Fortunately, the ICIEF method was intended to monitor the charge variant distribution, and it's not crucial to report the accurate PI, so the method still serves its purpose well. For all conditions tested, the electrophoregrams were assessed manually, and the one showing best resolution was selected. Here shows the electrophoregram after DOE study. The previously unresolved shoulder peak is now better resolved. The data analysis was performed in CampStation back then and now has been routinely performed in the Campus 2.0 software. To summarize, the PI shift observed during method transfer resulted from the discrepancy in method parameters. A DOE study was performed. The observation in PI shift was reconstructed with change in formulate composition. In addition, the ICIEF method was further optimized. In the second part of the talk, I will share a story about CESDS method validation. For another IgG1 molecule, both non-reduced and reduced CESDS assays were developed on the Maurice as a purity assay for release and stability testing in QC. The methods were almost identical to the default Maurice CESDS methods. These methods were transferred successfully to STC Biologics and validated with combined efforts of STCs and the client. Here it shows the electrophoregrams of both non-reduced and reduced CESDS. The method validation is designed in compliance with the ICH guideline Q2. Several typical validation characteristics were listed in the guideline, including accuracy, precision, specificity, detection limit, quantitation limit, linearity, and range. Besides, we also tested the robustness and onboard stability. Both non-reduced and reduced CESDS methods passed the specificity and accuracy criteria nicely. For specificity, there was no interfering peaks observed for the blank or formulation buffer. For accuracy, the back-calculated protein concentration matched the theoretical concentration nicely. However, both non-reduced and reduced CESDS assays didn't pass the linearity criteria. The proposed linearity criteria was to achieve no less than 0.99 of the coefficient of determination at protein concentration between 0.6 to 1.4 mg/ml. As is shown here, when plotting the peak area of intact IgG, or heavy and light chains against the protein concentration for non-reduced or reduced conditions, we only get an R-square value between 0.93 to 0.95. Visually, there is also curvature in the plot, which may result from signal saturation at high protein concentration. Following further consideration of the method purpose, the DSDP specifications and method performance requirement, it was determined that the linear range can be narrowed to a protein concentration between 0.8 to 1.2 mg/ml. Besides, since the reportable results are relative percentage area rather than absolute concentration, the linearity requirement needs only to show a proportional response for the corrected peak area versus sample concentration, and therefore an R-square value of no less than 0.95 can be justified. By narrowing the linearity range down to 0.8 to 1.2 mg/ml and modifying R-square criteria to no less than 0.95, both non-reduced and reduced CESDS methods pass the linearity criteria. Here shows other assay characteristics. We observed great repeatability and day-to-day, analyst-to-analyst, and lab-to-lab reproducibility. The relative standard deviation observed was no more than 2.8%. The limits of detection and quantitation were also assessed. For a non-reduced condition, the limit is set based on the detection and quantitation of major impurity, which is heavy light fragment. For reduced condition, the limit is assessed based on the light and heavy chain peaks. As a result, the detection limit was 0.1 mg/ml and 0.003 mg/ml for non-reduced and reduced CESDS. The quantitation limit is 0.2 mg/ml and 0.006 mg/ml for non-reduced and reduced CESDS respectively. The working range of both CESDS assays was defined by linearity, accuracy, and precision criteria. As was mentioned before, the range was narrowed down to 0.8 to 1.2 mg/ml due to the possible signal saturation observed at high protein concentration. Besides, both non-reduced and reduced CESDS showed great robustness with deliberate change in sample denaturation condition. Good onboard stability was also observed, that the samples post-preparation were found to be stable for up to 48 hours in a temperature-controlled sample compartment. To summarize, I shared two studies about ICIEF method transfer and a CESDS method validation. At STC, we routinely perform both ICIEF and CESDS testing on Maurice system to support product development and QC testing. Last but not the least, I want to thank my supervisors, colleagues, and our clients, who in collaboration put together the network presented here. I'd also like to thank the staff from Protein Simple for helping us to make the best use of Maurice. Thank you all for attending the webinar, and I will take questions together with Krisa after the next talk.
Thank you. Today, I will be presenting how my team and I developed a method for the separation of light chain isoforms by reducing CESTS using the Maurice instrument for one of our molecules. I'll begin with a little bit of background information of our molecule, including capillary isoelectric focusing, or CIEF, and mass spectrometry, or MS. Then I'll move on to reducing CESTS screenings on the Agilent 7100 instrument versus the Maurice. Then I'll go into method optimization on the Maurice, and finally end with some concluding statements. The molecule that we tested is a monoclonal antibody. One of the first assays that we performed on this molecule was CIAF, which resulted in the following electrophoregram. This peak profile is for the intact mAb and demonstrates a unique charge distribution compared to other mAbs that we have encountered previously. There is a large presence of acidic species making up about 78.2%. From here, we knew that there was a large amount of acidic species but did not know where this acidic charge was coming from. We decided to go further and do some more CIF analyses to determine where this charge could be coming from. We decided to treat our molecule with a sialidase, which cleaves any sialic acids present on our molecule. The green trace in this overlay represents our unmodified molecule, while the blue trace represents our molecule treated with a sialidase. From this overlay, we can see that once our molecule was treated with the enzyme, all of the acidic species resolved into one small acidic peak. From this data, we determined that the acidic charge from this molecule was coming from sialic acid. To determine the location of these sialic acids, MS data was generated for our molecule. All of these panels represent different manufacturing cell processes for the same molecule. From this data, we determined that the sialic acids are located on N-linked glycans on the light chains of our molecule. Most of these panels represent ideal cell culture processes because they yielded high molecular weight light chain species. Panel D is an example of a non-ideal cell culture process because it resulted in mostly low molecular weight light chain species. Sialic acids have critical functions for many biomolecules, including half-life of the molecule and clearance of the molecule in vivo. With this being such a critical attribute to our molecule, we needed a quick assay to be able to monitor the sialylation on the light chains of our molecule. We decided that the best way to monitor sialylation on our molecule was by performing reducing CESTS on our Agilent 7100 instrument. For parameters and conditions, we diluted our sample to a final concentration of 0.75 mg per ml. Our samples were prepared in a 100 millimolar tris HCl buffer with 1% STS and a pH of 9. This is the buffer that comes in the Beckman-Coulter CESTS kit. Our alkylating agent was IAM, and our reducing agent was BME. For all methods on our Agilent instrument, we implement a step injection. This was recommended to us by CESTS experts so as to not shock our molecule with such a high voltage. For this step injection, our final voltage was 10 kilovolts and lasted for 22 seconds, and the separation had a voltage of 16.5 kilovolts for 50 minutes. With preconditioning and postconditioning, each injection lasts for around 65 minutes. An IgG control was also analyzed in this batch for system suitability. Displayed here is the reducing CESTS profile of our molecule on the Agilent 7100 instrument using the parameters and conditions previously outlined. The molecule is fully reduced with a small presence of impurities. After integration with the Agilent ChemStation software, a percent purity value of 97.4 was determined. Looking more closely at the electrophoregram, we can see that the light chain separation shows a great distribution of end-linked glycan variability on our molecule. The multiple light chain peaks that are present correlate to different amounts of sialic acid in various locations on the light chains of our molecule. At this point, we deemed this method acceptable to monitor purity of our molecule and the glycan variability, and we were ready to begin qualification. Upon receiving the Maurice, we decided to see if we could perform reducing CESDS for our molecule and obtain comparable results to what we got on the Agilent 7100 instrument. For the Maurice screening, we went with all Maurice CESDS reagents, default methods, and conditions. We diluted our sample again to a final concentration of 0.75 mg per ml and used BME as a reducing agent. For the default Maurice reduced IgG method, the injection parameters were 4600 volts for 20 seconds, while the separation parameters were 5750 volts for 25 minutes. Compared to the Agilent 7100 instrument, the voltage on the Maurice is much lower and the separation time is half of that. We did not know what to expect, but were hoping that the results would be comparable. An IgG control was analyzed for system suitability for this batch and each batch going forward. Compass software was used to analyze all Maurice data. With the Maurice default parameters and conditions, this is the electrophoregram that was yielded. The molecule was well separated, but what we initially noticed was that the overall signal was much higher than the recommended range by ProteinSimple. After integration, we obtained a percent purity value of 98.2, which is in fact slightly higher than what we obtained on the Agilent instrument. Taking a closer look at this electrophoregram, we can still see that there is a very small presence of impurities, but the light chain peaks are not nearly as well resolved as what we got on the Agilent instrument. At this point, we decided to move forward with method optimization in hopes to decrease overall signal and increase resolution of light chain peaks. Our first step in optimization for the reducing CESDS method on the Maurice was to adapt our Maurice method to what we currently do on the Agilent 7100 instrument. The first thing we did was to incorporate our step injection and increase the final voltage for all samples, excluding the IgG controls. From here, we implemented the step voltage and ended with a final voltage of 6500 volts. This is the highest that the Compass software would allow us to go. We also wanted to compare preparation of our molecule in the 1x Maurice sample buffer versus the Beckman-Coulter sample buffer. In this overlay, the blue trace represents our molecule with default Maurice method parameters, while the green trace represents our molecule with the newly implemented step injection and increased final voltage. With our new parameters, the signal overall has increased, which is what we tried to avoid initially, and although the light chain peaks do have slightly more resolution, it is still not optimal. After reading the Maurice CESDS development guide, it was recommended by ProteinSimple to not increase the voltage by too much because this could induce Joule heating. Joule heating can affect your molecule depending on the different components in your sample matrix, and we wanted to avoid this at all costs. In this overlay, the blue trace represents our sample prepared in the Maurice 1x sample buffer, while the green trace represents our sample prepared in the Beckman-Coulter sample buffer. There's no major differences in peak profile between these two buffers, so we decided to continue with the Maurice 1x sample buffer since it's what is recommended by ProteinSimple and it is what is compatible with the Maurice instrument. For further optimization, we decided to keep the step voltage implemented, but decrease the final voltage to 4600 volts to avoid any joule heating. We also wanted to test decreasing final injection voltage and decreasing total injection time. CESTS uses an electrokinetic injection. Therefore, when the capillary inlet is submerged into the sample and voltage is applied, any charged ions, which means your protein or any charged buffer components, will enter the capillary. Decreasing injection voltage should inject fewer ions and in turn increase resolution and decrease overall signal. With decreasing injection time, there is less time for the sample to migrate into the capillary, therefore increasing resolution and decreasing overall signal. In this overlay, the blue trace is our molecule with default reduced IgG conditions, while the green trace is our molecule with the step injection ending at 4600 volts. While both profiles are very comparable, the step injection induced slightly better resolution of the light chain peaks. Since there were no negative effects, we decided to move forward with the implemented step injection. This is an overlay of decreased injection voltage. From what we can see here, as the injection voltage is decreased, the overall signal is decreased to well within the recommended range by protein simple. Looking a little more closely at this electrophoregram, we can see as the injection voltage is decreased, the resolution of the light chain peaks is increased. What started off as two main peaks with a couple of broad shoulders are now turning into defined light chain peaks. Thus far, the 3500 final voltage for injection yielded the best light chain peak separation on the Maurice thus far. This overlay displays injections with decreasing injection time. Like decreasing injection voltage, when the injection time is decreased, the overall signal is decreased to within the recommended range. We can also see that as injection time is decreased, the resolution of the light chain peaks is increased. Looking more closely at this overlay, we can see once again that what started off as two broad light chain peaks with a couple of shoulders have resulted in extremely well-defined light chain peaks, which are very comparable to what was initially obtained on the Agilent instrument. After viewing this data, it was clear that a 10-second injection time, which is the red trace, yielded in the best separation of the light chain peaks on the Maurice thus far. Just to compare the two most optimal conditions, we can see that the 10-second injection time still resulted in better light chain separation than the decreased injection voltage to 3500 volts. So to summarize our optimal conditions and parameters for our Maurice reducing CESTS method, for sample preparation, we will be using Maurice 1x sample buffer and BME as a reducing agent. And for method parameters, we will continue to implement a step injection with a final voltage of 4600 volts for 10 seconds and a separation of 5750 volts for 25 minutes. This overlay compares our molecule with the default Maurice reduced method versus our optimized reduced method. From this overlay, we can see that our optimized conditions and parameters resulted in a considerably lower signal that is well within the protein simple recommended range. By zooming in, we can see that our optimized method parameters and conditions result in very well separated light chain peaks that clearly demonstrate the glycosylation variability of our molecule. Once a repeatable profile for our molecule was achieved, we had to determine optimal analysis settings for auto-integration. Our Compass software is CFR compliant, which does not allow for much manual integration of our electropherograms. With default analysis settings, our light chain peaks are divided into two main groups with auto-integration, and not many small impurities are detected. To achieve proper auto-integration of our molecule, we adjusted the analysis settings peak find width from 35 to 10, which resulted in the following profile. Once the analysis settings were adjusted, we were able to properly integrate our light chain peaks and shoulders and detect all small impurities. After integration, a percent purity value of 98.3 was determined, which was very comparable to what we got on the initial screening, meaning we did not lose any purity, and it is still higher than what we achieved on the screening using the Agilent 7100 instrument. When comparing the Maurice versus the Agilent 7100, both peak profiles are very similar. Although both instruments can be used for reducing CE-SDS of our molecule, we were able to achieve optimal resolution in half of the time with the Maurice. There was also a more steady and consistent baseline with the Maurice, which allows for easier auto-integration and better detection of small impurities. Optimal light chain separation was possible, and percent purity was slightly higher with the Maurice. Despite having CFR-compliant software, we were able to achieve analysis settings that properly integrated our molecule. In conclusion, the Maurice is extremely easy to operate and is still very comparable to other CE-SDS instruments. Optimization of our molecule was mostly through modified method parameters, which means less variability and less manipulation of your molecule. Lastly, it was found to be a suitable method for release and stability in QC. I would like to thank all of the people on my acknowledgements page for all of their help and support throughout this entire process. Thank you for your attention, and I will be happy to answer any questions during the Q&A session.
I would like to thank everyone who joined us today for this webinar. If you have any further questions, please feel free to reach out to me. My name and my email address is displayed on the screen. On behalf of Pretty Simple and Cambridge Health Tech Institute, I would like to thank our panelists, Dr. Chen Liu and Chris Edwards. Thank you, everyone, and see you next time.